Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. Now, in the previous video, we began our exploration of genetics and talked about the beginning stages of Gregor Mendel's experimentation that ultimately laid the foundation for this entire field of study. And the law we discussed was the law of segregation. Now, this week, we're going to move forward and discuss the law of independent assortment. Right now, you should be seeing the terms and definition slides popping up so that you can pause the video and grab the vocabulary that we will be using in this discussion. A quick note that the terms and definitions are going to be the same as in the last video because the material we're covering uses a lot of the same terminology. So if you have the vocabulary from the last video, you have the vocabulary for this video. A quick reminder though, if you would like my entire slideshow presentation in PDF format to help you study or help you learn this material, please pop on over to my Patreon where you can grab that PDF for absolutely free. However, patrons do get this content at least a day in advance. All right, with that being said, let's do a quick review of what we went over in the last video. So first off, genes are carried on chromosomes at specific loci, an example being the gene for flower color. And then alleles are alternate versions of these genes, an example being the allele for purple flower color and the allele for white flower color. So it's the same gene, just different versions. And then organisms carry two alleles for each gene, one being inherited from each parent organism. A homozygous organism has the same allele for that gene, so it's the two copies are the same, whereas a heterozygous individual has different alleles for one gene, so their two alleles are different. A dominant allele will determine the phenotype. So in the heterozygous organisms that have those different alleles, if one allele completely masks the other, so only that phenotype is seen, then that allele is dominant and the other allele is known as recessive. And in this case, a recessive allele can only be seen in the phenotype if the organism is homozygous for that allele. A sperm or an egg will only carry one allele for each gene. So during meiosis, when those homologous chromosome pairs separate, the sperm or egg that results at the end will carry one allele for each gene. And then a Punnett square can help visualize the possible phenotypes and genotypes of offspring given the genotype of the parent organisms. So last week, we looked at the famous experiment that had to do with flower color, where that law of segregation was discovered, that the particular alleles for each organism will segregate independently during meiosis, and the sperm and the egg will carry one allele. So these genes can be shuffled and all that fun stuff. But what we were looking at is known as a monohybrid cross. And this is basically an experiment where organisms that are heterozygous for the one character of interest are bred, or in the case of flowers, self-pollinated. And the trends in that offspring that result from this breeding are the results of the experiment. So the example here being the flower color experiment, where the F1 generation was heterozygous for the purple and white flower color, and then those plants were bred to get that F2 generation, where we had this lovely ratio of 3 purple flowers to 1 white flower for the offspring that resulted. And we learned about the difference between the phenotypic ratio and the genotypic ratio. But can we observe more than one character at a time? Can we only do monohybrid crosses, or is there a way to look at multiple different characters in an experiment like this? Well, the answer is yes. We can do these experiments where we're looking at multiple characters. And an experiment that we're going to look at today that Mendel did is known as a dihybrid cross. And like the monohybrid cross, this is an experiment where organisms are bred, but the difference here is that the organisms are heterozygous for the two characters of interest. And then again, the trends in the resulting offspring will be the result of that particular experiment. The one we're going to specifically look at is going to be the experiment Mendel did with seed color and seed shape. So seed color, the yellow color is going to be the dominant trait in this case, and the green color is the recessive trait. And then in seed shape, the dominant trait is that smooth, round shape, and the recessive is that more wrinkled shape. So the first hypothesis that they looked at when they originally did this experiment was the hypothesis of dependent assortment. And this hypothesis essentially says that the alleles for seed color and shape will be inherited together. So the P generation would have had the homozygous dominant in both traits and the homozygous recessive in both traits. Then they would have been bred, gotten that F1 generation that was heterozygous for both traits across the board. And the expected results here would be a Punnett square that looks exactly like the one of the flower color, where we get that 3 have that dominant phenotype and 1 has the entirely recessive phenotype. So what we're thinking here is that if you have the dominant trait, you're going to pass on the dominant trait in both characters. So in this case, the yellow color and the smooth round shape. So those would be inherited together, and same with the recessive. That green color and the wrinkled shape would always be inherited together. But is this what they saw? Is this what Mendel observed when he did this experiment? No, it's not really what Mendel observed. So again, here we see a graphic of what they were expecting to see. The Punnett square that basically would mirror that of the flowers with 2 phenotypes, the round yellow and the wrinkled green, in the 3-to-1 ratio. But again, that was not what was observed. So that brings us to the second hypothesis, which is the hypothesis of independent assortment, which says that genes are going to be inherited independently of one another. So the Punnett square we see here on this slide is the one that's looking at 2 traits that are inherited independently of one another. So this F1 generation, remember, it has that double heterozygous for both of those traits. You have to then look at the possibilities that each sperm or egg can have from this individual, meaning there are 4 possibilities. It can either pass on 2 dominant traits, it can pass on the recessive for color and dominant for shape, the dominant for color and the recessive for shape, or pass on both recessive traits. So this extends that Punnett square out to be 16 possibilities. And the results that you see, you have 4 distinct phenotypes. So that round yellow, the round green, the wrinkled yellow, and the wrinkled green that represent all of those possible genotypes and allele interactions that we're seeing here. And the ratio that you're going to get from an experiment like this in general is going to be a 9-to-3-to-1 ratio overall. So this brings us to what is now known as the Law of Independent Assortment, which says exactly that. The pairs of alleles are going to segregate independently of each other during meiosis, and you're going to, in general, get that 9-to-3-to-3-to-1 ratio in these dihybrid crosses. And Mendel did confirm this with various tests where he looked at different combinations of these 2 characters. And then the big thing here is that the inheritance of one character is going to have no impact on the inheritance of another character. So we're going to keep seeing these 16 square Punnett squares with that ratio. And the thing to remember here is that 9:3 to 3:1 ratio. If you get into an upper level genetics class, that particular ratio is one of the ways that you recognize specific laws and specific interactions and specific things with this complete dominance and independent segregation. A quick recap here of Mendel's laws. So the law of segregation. During meiosis, alleles will segregate and each gamete carries one allele for each gene. The law of independent assortment is that during meiosis, the alleles for one gene are going to segregate independently of the alleles for other genes. Now, of course, there are going to be exceptions, and the rules do get modified depending on what we're looking at and what we're observing, but these are going to be the general rules that provide the foundation of what we know in the field of genetics. But we're not going to get into those exceptions today. We will cover that in future videos. Before we really move on with genetics, we're just going to touch on probability for a moment, because Mendel's laws are seen to reflect probability, and the two big rules here are the rule of multiplication and the rule of addition. Now, I'm not really going to get into the nitty-gritty of these rules, but in general, to help you know what you need to do when you receive a question on a biology exam, if there's only one way to get the result that you're looking for, or there is an and in the question, then you're going to multiply those individual probabilities. So looking at this Punnett square where we're looking at the flipping of coins to represent the sperm and the egg, if you're looking for that double dominant, so that homozygous dominant trait, there's only one way to get it. And in order to get that, the sperm has to have that dominant trait and the egg has to have that dominant trait. And when you look at those individual probabilities, you see that there's a 1/2 or 50% chance that the sperm is going to get that dominant trait. So 1 in 2. And same with the egg. There's a 1 in 2 chance that the egg will end up with that dominant trait. So to get the actual probability of getting that homozygous dominant offspring, you will multiply that 1/2 times 1/2 to get the 1 in 4 chance that you get that homozygous dominant offspring. Now, the rule of addition is what you would use if there's more than one way to get the result that you're looking for, or there is an or in the question. So looking at the Punnett square, a very simple example here, we want to know the probability of getting a heterozygous offspring. Now, if we look at the squares, we see that there are two possible ways to get a heterozygous offspring. Either the sperm can donate the recessive trait, or the sperm can donate the dominant trait. And then same with the egg. The egg can donate the recessive trait, or the egg can donate the dominant trait. So for each of these individual occurrences, the probability is 1 in 4. But to get the total probability for getting the heterozygous offspring, you have to now add those 1 in 4 probabilities together. So the overall probability, once you add 1 in 4 to 1 in 4, is going to be 1/2. So there's a 50% chance to get that heterozygous offspring. We're not going to get into it more than that. We are going to move on at this point. But again, the big thing to know if you need to multiply or add is if there's only one way to get the result, you're probably going to use the rule of multiplication. And if there's multiple ways to get the result you're looking for, you're probably going to need to use the rule of addition. What if you don't know the genotype of an individual? How could you possibly figure out what that individual's genotype is? Especially if they're just showing that dominant characteristic. Well, one thing you can do is you can do a test cross, which a test cross is basically an experiment that helps you determine the unknown genotype of an organism by breeding them with an individual with the known genotype. And how you're going to do this is the unknown organism is going to have that dominant phenotype. So you don't know if it's homozygous dominant or if it's a heterozygous individual. Because remember, when you have a dominant allele, if the individual is heterozygous, you're going to get that dominant phenotype. And then the known phenotype, you're always going to have the recessive phenotype. And that's because it's going to be homozygous recessive. And if it's a dominant, then you still have a lot of questions, because if all of the offspring have the dominant phenotype, you don't know if it's because they're both homozygous dominant or if it's because the unknown individual is heterozygous, and because you bred them to a homozygous dominant, you're always going to get all dominant phenotypes. So it's going to be an unknown with the dominant phenotype and an unknown with that recessive phenotype. Therefore, the genotype is going to be a homozygous recessive. In the example that we're using, we're going to be looking at black and chocolate labs. Now, with the lab dogs, the black coloration is the dominant trait. So the dominant phenotype is the black coat color, and the recessive is that chocolate color. And here, our unknown is this black lab. So we know it has at least one dominant allele because it's black. And then our known is that homozygous recessive chocolate lab. And what this test cross is going to do is it narrows it down to the two possibilities for that black lab. It's either going to result with all black puppies, meaning that the black lab has to be a homozygous dominant, because if it's a heterozygous dominant, you're going to get 50-50 black puppies and chocolate lab puppies. And it's as simple as that. You just breed them and look at the results. If you have the entirety of that result being the dominant phenotype, you have a homozygous dominant result. If you have about a 50-50 ratio, or honestly even one with the recessive phenotype, you know you have a heterozygous unknown. Now, a little disclaimer here, because genetics is messy, because things are entirely random when you breed, it would be possible to get the all black, but actually have the heterozygous be the unknown. They just happen to not pass on that recessive trait in this one breeding. However, it is unlikely. And so this is going to move us on to how we look at genetics in humans a little bit. And one way that we track the movement of particular traits and phenotypes in humans is by using a pedigree. And a quick way to read this pedigree that we're currently looking at is that the shape is going to denote whether the individual was female or male, and then the colored in or lack thereof denotes which trait they have. This particular pedigree, we're looking at the two traits of having either a widow's peak at the hairline or a completely straight hairline. So the widow's peak is the dominant trait, and this is denoted by these kind of open circles or squares. And the straight hairline trait is that recessive trait that is denoted by the filled in circles or squares. And the way that we're going to use these pedigrees, especially if we don't know if certain individuals are heterozygous or homozygous for the trait that we're looking at, is we'll look at the offspring, so the children, and trace it back through the lineage. Because, for example, if we're looking at this particular pedigree at that third generation, the individual Kristen that has the straight hairline, we know has to be homozygous recessive, because that is the recessive of the two traits. So going back, we see that both of her parents have a widow's peak, the dominant trait. But because she has that recessive trait, we know both of her parents have to be heterozygous for that trait. And you can go back further and figure out the particular genotypes for the individuals for many generations. but that's the general way of how you look at it. It's very much like a family tree, just instead of only looking at who's related to who, we're looking at what traits are present and where they pop up in that family tree. So you basically use the children to figure out the genotypes of the parents if the parents don't have that recessive trait being expressed. So that brings us to the question, is dominant the norm? Are dominant traits always going to be the most common trait in a population? And what we see is that no, dominant traits are not always going to be the most common trait in a population. So in this image on the slide, we see two traits, freckles and the albinism traits. Now, the dominant traits in these two cases are the presence of freckles and having pigmentation in the skin cells and hair cells and all that fun stuff. And the recessive traits are going to be having no freckles and having albinism. And we can see here by the coloration which one is more dominant in populations. So in the case of freckles, having no freckles is going to be more common, but in the case of albinism, having pigment is going to be more common. We can see here that not always having the dominant trait means that it's going to be the more common trait, because, like I said, the freckles here is denoted as being less common. And how we refer to that less common trait is as the mutant trait. Now, this is just the way that we refer to it. It doesn't necessarily mean anything negative. It just denotes that this trait is the less common trait in the population. And then it's important to kind of have these pedigrees and kind of understand what's going on here, is because there are recessive disorders like albinism that do range in severity. So albinism would be known as a mild recessive disorder, and something like Tay-Sachs disease is a fatal recessive disorder. And if we have these pedigrees, we can track where the different traits are in different lineages and all that fun stuff. And if we have these heterozygous individuals that have that recessive allele, but it's not expressed because, again, it's recessive, so we get that dominant expression, they're known as carriers. So they have that recessive allele and they can pass it on. It's just not expressed in them because they also have the dominant allele to cover it up. And then it's also important to note that these autosomal disorders, so like albinism and Tay-Sachs, they can be either dominant or recessive. Just because it's a dominant trait doesn't necessarily mean it's not a disorder. And again, disorder here is just the way that it's going to be described in this academic sense. In certain conditions, it doesn't necessarily mean anything's wrong with you, and for the most part, you can be healthy otherwise, minus that little trait that's just not common in the population. And I don't mean to imply anything. It's just how it's going to be referred to in the science classroom. So here is a lovely little table from the book that just has some of those recessive and dominant disorders for you to look at. Most entry-level bio classes aren't going to get very in-depth in any of this. Just kind of know how to look at that pedigree, know how to kind of read it and interpret it, and know that dominant is not always most common. And if we're looking at disorders, they can come from either a dominant or a recessive allele. So that's where I'm going to leave you with this video. Have a wonderful rest of your day, afternoon, evening, what have you, and I will see you all in the next one.
Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.